Everyone. Now we'll begin with the first technical session of the day. We are honored to have with us Professor Zofia Shwikowska-Kulinska, who is currently working at the Department of Gene Expression, Adam Mickiewicz University, Poland. She did her PhD in transfer RNA genes in wheat from Adam Mickiewicz University in 1986. She has supervised multiple doctoral theses MSc students, bachelor students, etc. She headed the Institute of Molecular Biology and Biotechnology at Adam Mickiewicz University for 19 years. She built up IMBB, recruited many excellent scientists, and opened up new avenues for research like human molecular genetics and bioinformatics. She has coordinated multiple research grants funded by the Polish national agencies, one EU project, and participated in multiple others. She was head of the National Research Leading Center project at the level of Institute of Molecular Biology and Biotechnology, awarded by the Polish Ministry of Science and Higher Education. She has won many awards such as Polish Genetic Society for the best paper published in plant genetics in 2012-2013, AMU Rector Fellowship for Scientists with the highest score of publications in 2012, 2013, 2014, Award of the Polish Academy of Sciences 2014, the Lifetime Achievement Award from the Polish Ministry of Science and Higher Education 2016, and many more. She is a member of EMBU and many other scientific bodies such as ERA CAPS Advisory Board, Polish Central Commission for Degree and Titles, Council of Provosts for Division 2 of Polish Academy of Sciences, Scientific Council of the Institute of Plant Genetics, PS. Her main research interests include RNA metabolism, microRNA biogenesis in plants, and its regulation in various abiotic stresses. Crosstalk between spliceosome and microprocessor machineries in microRNA biogenesis, role of microRNAs in sex organ development in liverworts, and various strategies of potato varieties in response to drought. So with that, I would like to welcome Professor Zofia.
Okay. So thank you very much for coming. Can you hear me? Yes? Okay. So we will start today. This is an introduction to regulatory molecules like microRNAs within GIAN project, as we already learned today in the introduction part. So we should start with the best message for microRNAs from the last year, that both guys, this is Victor Ambros and Gary Ruvkon, they work in the University of Massachusetts, Chen Medical School, Worcester, USA, and Harvard Medical School, Boston, USA. They got both a Nobel Prize for the discovery of microRNA and its role in post-transcriptional gene expression. So what did they do? We will start a little bit historical. Everything happened in 1993, and I had the pleasure in 1994 to attend a meeting where one of them, I do not remember now who of them, gave a lecture on these small RNAs, those days called short temporal RNAs, stRNAs, because they appeared during C. elegans, Semiratitis elegans life cycle, shortly, very shortly in a precise time. That is why they called them short temporal RNAs. So this is C. elegans, Semiratitis elegans, is a worm belonging to nematodes. And what is its life cycle? Is it visible in these slides? Okay. So we start with an adult who is hermaphroditic, means that it has male and female parts. And after fertilization, the zygote undergoes multiple divisions for 14 hours. Then you get the larval stage 1. Then the larval stage, after 7 hours, turns to N2, to N3, to N4, then young adult, and finally adult, able to give rise to other generations. Eventually, when it is too crowded, or there is enough food, at the stage of N1 to N2, the larva can go to the so-called pre-larval stage, and then to the larval stage, and stay there for a couple of months. And then when the environmental conditions are again advantageous, it can directly turn to N4 stage. So what both guys, Gary Ruvkon and Ambros, did, they short temporal RNAs called microRNAs. So these were two papers published in Cell in 1993, back to back, so together, and then additional one that revolutionized the RNA field, published in Nature in 2000, about the other short temporal RNAs, already called microRNAs, but the discovery was that, that this is also present in other animals. Because in 90 years, nobody was so really interested in these short temporal RNAs. Everybody said, well, this is worms, some very specialized way how to control larval stage transitions, nothing interesting for other animals. And then suddenly Gary Ruvkun discovered that one of these short temporal RNAs is present in human genome after sequencing of human genome. And then he discovered that it is present also in other animals, from invertebrates to vertebrates. And this revolutionized the field because it meant that if something is kept during the evolution from worm to human, it means something, that it must be important. So what did they do? They discovered this Lin4 microRNA that controls transition from larval stage 1 to larval stage 2 via downregulation of Lin14 and Lin28 mRNAs. So here you see the wild type of the worm, here you see KO mutant of Lin4 and KO mutant, knockout mutant, yeah? KO mutant for Lin14 mRNA. So the Lin14 is the most severe phenotype of the mutant. So what is the role of Lin4? So Lin4 has its own gene that is not expressed until late L01 stage. Then the repressor that is normally occupying this promoter region is degraded and the transcription starts. On top of this, what is interesting and rather unique until now, there is a place in the promoter region complementary to small RNA, to this Lin4 microRNA, and Lin4 hybridizes within promoter region, stimulating its transcription. On top of that, RBM, RNA binding protein 28, stimulates microRNA biogenesis, so you have a lot of microRNA Lin4 at the late stage of L1. So, of course, I will put it on. Is it fine? Yeah. So, what microRNA is doing? This is mRNA. This is its 3' UTR. 3' UTR, 3' untranslated region of mRNA. So here is a coding sequence. Here is its 3' UTR. And you see there are many, many sequences complementary to the Lin4 microRNA that is occupying this 3' UTR region. And this binding leads to translation inhibition. So downregulation of Lin14. What is Lin14? Protein. This is a heterochronic chromatin that is responsible for the developmental patterning of animal formation. So these are really crucial proteins, transcription factors mainly, that are responsible for particular developmental stage. And on top of that, there is Lin28, that is one place where Lin4 can bind, and Lin28 is stimulating the activity of Lin14. So both have sequences that are complementary to Lin4, and thus both mRNA's translation is inhibited, resulting in downregulation of Lin14 and Lin28 protein production. And this leads to the transition to L2 larval stage. And the second one, LET7. LET7 controls the transition from the larval stage L4 to adult form via downregulation of Lin41. So you have a LET7 microRNA and Lin41 that is again a heterochronic gene and again transcription factor. And in Lin41, you have two binding sites for LET7 in its 3' UTR. Here are exact sequences for complementarity. And when LET7 is expressed, and it is expressed late in L4 stage, it downregulates LIN41. What means when it is downregulated, LIN29 that is transcribed by LIN41 is upregulated, right? When we have a lot of LET7, LIN41 goes down, and it means that we have a lot of LIN29, what means inhibition of all events within L4 phase, larval stage, and it opens a way to the development of adult events leading to the adult phase. So we have two microRNAs regulating L1 to L2 transition and L4 to adult stage. Is it clear?
Yes, please.
Yeah. So these were the first microRNAs discovered, and since then the field was there emerged a new field of microRNAs, and everybody jumped on it, of course, because it is something new, it's that hot topic, and you can generate good papers working on new discoveries. So what do we know about microRNAs? They represent small RNA molecules, usually 20 to 22 nucleotides in length, and they act as pivotal regulators in post-transcriptional gene silence. In plants, they were discovered in 2002. It was a famous paper from David Bartel-Gros, published in Genes and Development. And when it was clear that we have microRNAs also in plants, we have microRNAs in animals, we have practically everywhere microRNAs. This is a question of fungi. We know that Saccharomyces cerevisiae does not have microRNAs. There are some groups of microRNAs in other fungi, but it is still not very clear. So they are in animals and they are in plants, but they have some differences typical for both, let's say, kingdoms of animals and plants. So microRNAs influence the fate of transcripts that contain sequences complementary to microRNAs in two different ways. They may lead to mRNA degradation, or they may lead to translation inhibition. So the first way to leading to mRNA degradation is typical for plants. Is it possible then to have these slides a little bit up, because it is it will not help. This is in the screen, I think. That I don't know how to do.
Off here.
Maybe we can do it up a bit, maybe.
You have to edit the slide, ma'am, I think, for that.
Like this, maybe I will do
Yeah.
the whole, where it is. No. Okay, the whole slide. Okay. I will put this up. Maybe not. I will do it for all
The figure
other.
is, I think, too low.
And so now is better? Is better? No, wait a moment.
Control Z.
Control C. So can we go back here? Yeah. Is it now good?
It's in the
Yeah. So this is the mode typical for plants. So microRNA is embedded in a complex called trace, RNA-induced silencing complex. The main executor of the complex is a protein that is a member of Argonaute family. In plants, this is Argonaute 1. We have 10 Argonauts in Arabidopsis, but Argonaute 1 is responsible for the majority of microRNA's action. So microRNA embedded within this RISC complex is guiding this RISC complex to a complementary mRNA. AGO1 protein is a nuclease, endonuclease, that cleaves mRNA just in the middle of hybrid of microRNA and mRNA. as a result, you get two halves of mRNA, and both halves are then degraded by exonucleases 3', 5', 5', 3'. Yeah? And this is the fate. The majority of microRNAs act like that in plants. While in animals, the preferred mode is, and one more thing, sorry, that the majority of microRNAs in plants bind to coding sequence. Yeah? While in animals, it binds to 3' UTR in the majority of cases, and upon binding to 3' end of mRNA, it inhibits translation. But finally, it also leads to mRNA degradation, because what happens after translation inhibition is the poly-A tail degradation, then decapping of mRNA. You are aware that mRNA has cap and poly-A tail, right? Yeah, so decapping means the removal of cap structure, and deadenylation means the removal of poly-A tail, and then mRNA is easily degraded. So in both cases, maybe the mechanism is different, but it leads to degradation of mRNA, sometimes in animals in depositions in so-called P-bodies, but in the majority of cases, it is degraded. Okay, so now microRNA biogenesis in animals, how does it? So we have here nucleus in humans. This is transcript produced by RNA polymerase II, and it contains a specific stem and loop structure. And within the stem of the stem and loop structure, microRNA and microRNAs are embedded. DROSHA and DGCRH2 proteins are responsible for producing just stem and loop structure that, bound by exportin 5, is exported to the cytoplasm, and now exportin 5 releases stem and loop structure that is now a substrate for another enzyme called dicer that now cleaves stem and loop structure to a duplex of mature microRNA and passenger strand. And now this is recognized again by RIS, of which AGO is AGO2 mainly, or AGO1 in human, is main executor for binding of microRNA. One strand, passenger strand, is removed, and the AGO-RIS complex remains with microRNA ready to act on cytoplasmic mRNAs. The whole action of microRNAs takes place in cytoplasm. Yeah? They act on mature mRNA. So how in plants? Sorry, I should do another way. So I have no movie for plants, unfortunately. So this is the nucleus. This is cytoplasm. So the gene is again transcribed by RNA pol 2. In general, all microRNA genes are transcribed by RNA pol 2. In animals, we have several examples where RNA pol 3 is transcribed with microRNA genes. And in plants, we know only one example, published quite recently by Michael Axel, a laboratory of some parasitic plants that probably are transcribed by RNA polymerase 3. This is the only example I know. So the gene is transcribed, you get a transcript with this classical stem and loop structure within it, where in the stem microRNA and star are embedded, and this is immediately bound by so-called microprocessor. And microprocessor is a huge complex, but the core proteins of microprocessor are DCL1, dicer-like 1 protein, this is the cleavage RNAs that will cleave mRNA. Then you have serrate, protein important for microRNA biogenesis, and IL-1 also important for microRNA biogenesis. So if you cannot get KO mutant of DCL1, this is lethal for plant. You cannot get serrate KO mutant, it is lethal for plant. You can get IL-1 KO mutant, but the plants are quite dead. So this is the core of microprocessor, these three proteins. And then dicer-1 cleaves stem and loop structure out. So you have here the stem and loop structure. Then dicer-1 again takes a second cleavage event, producing microRNA duplex with passenger microRNA. Here is an additional interesting event that 3' ends of passenger and microRNA are 3' O methylated, and it protects them from degradation. And then the duplex, or already one strand, it's still not clear, is embedded within a risk complex in which AGO1 is the main executor. And this is an export gene of microRNA from nucleus to cytoplasm called CRM1. And then it is exported to cytoplasm, where microRNA guides AGO1 to a given mRNA, and mRNA is then cleaved first in the middle of hybrid, but then further degraded. So, summing up the differences in microRNA biogenesis between animals and plants, we may say that in plants, practically all events of microRNA biogenesis take place in the nucleus, while it is divided in animals, that stem loop structure is produced in the nucleus, but then it is exported to cytoplasm, where it is cleaved further by another enzyme, here dicer, and in the nucleus dosha, to give rise to a duplex that then incorporated into this complex guides it to a given mRNA. And in plants, everything is going on actually in the nucleus, and AGO1, either with duplex of mRNA or already with a single microRNA, single-strand microRNA, is exported to cytoplasm, where it targets a given mRNA. Is it clear?
Yes,
Mm-hmm. Okay. So now there is also a difference between let's say, animals and plants in the hybridization rate. So in plants, mainly microRNA hybridizes at the whole length of microRNA to mRNA. Of course, here I show you 100% complementarity between microRNA and mRNA, what is not always true. You may have 3, 4 mismatches, even in some cases even more, but still binding. But generally, it really nicely hybridizes to mRNA. So in the case of plants, it is not so difficult to identify target of a given microRNA. But in the animals, only 5' end of microRNA hybridizes fully to 3' UTR of a given mRNA. And this is so-called, this is 5, 6 nucleotides of 5' end of microRNA. This is so-called seed sequence of microRNA. And it hybridizes to a given mRNA. What means that many mRNAs can be a target of a given microRNA, and it is not easy to predict targets for microRNA in animals, so you have to prove it experimentally. So now, so what about microRNAs in numbers? There is a NIR-based webpage where all microRNAs discovered are deposited from animals, from plants, from algae, also plants, but lower plants, algae. And nowadays we know that till now almost 2,000 microRNAs have been found in humans. More than 1,200 microRNAs have been found in mouse, but only 326 in Arabidopsis thaliana, and 604 in Oryza sativa. So why do we have not so many microRNAs in plants in comparison to animals? So there may be 2 reasons. First, maybe not all microRNAs in Arabidopsis oryza are discovered. But I wouldn't say it is possible, but it will never grow to, let's say, 2,000 from 300 or from 600. Yeah? So according to me, we have in plants different classes of small RNAs that may substitute for these low number of microRNAs. I mean mainly TACI RNAs, trans-acting small RNAs. You have non-coding long RNA that starts to be cleaved by microRNA, and then RNA-dependent RNA polymerase is producing second strand, and then dicer is cleaving it into 20 to 24 nucleotide long small RNAs that have a lot of targets. So this is maybe the way the reason why we have not so many microRNAs in plants. Your battery is running low. So what to do? Sorry for that. So what about microRNA functions? So microRNA are really important. Regulators. First of all, let us talk a little bit about development. So what we know that flowers' development depends on microRNA. These are several modules. And module, I mean microRNA and its target. Yeah? Several modules controlling flower development. I will talk a little bit deeper about some of them. Stem development, another modules that are responsible for stem development. For leaf development, also some modules responsible for leaf development, and the same for root development. And going in life cycle, you have the same. Let's start from embryo, seed, root cell. You have modules involved in development, and from juvenile phase through some maintenance, so short stem apical meristem, to juvenile to adult transition, to adult phase, and vegetative to reproductive transition, we have typical modules that are important for these transitions. The most famous is the transition between vegetative to reproductive transition, and I will shortly talk about it. So practically all organs and all developmental stages of plant life cycle are to some extent controlled by microRNAs. May I? Yes, ma'am. Okay. So now plant microRNAs are also involved in plant responses to biotic and abiotic stresses. So what about biotic stresses? So biotic stresses means pathogenic bacteria, fungi, insects, nematodes. So what we know, for instance, in Oryza sativa, if there is an infection by Magnoporte oryzae, this is a fungi, a pathogenic fungi, if you get increase of microRNA 7695 that has its own target, it will confer resistance and rise against the fungi. Or for instance in Arabidopsis, where roots can be attacked by nematode infection. I did something, I don't know what. By nematode infection, microRNA 396 is downregulated, leading to reprogramming of root, and it confers also some kind of resistance against nematodes. And also microRNAs regulate abiotic stresses responses like drought and salinity, and what we observe, we see differential expression of many microRNAs under salinity stress. We see a lot of microRNAs also differently expressed during drought. For drought and heat, we will have special lectures because these are threats, I think, also very important for India as well. In Poland nowadays, we become more and more dry country, and we were quite a rainy country, and now it's more and more long without rain periods. So they are important in both abiotic and abiotic responses in plant life. So what are the main targets of microRNAs? So plants' microRNAs have a strong propensity to target genes controlling development. And By that, they target transcription factors that regulate transcription of other genes, and F-box proteins, mainly components of ubiquitin ligase complexes, so leading to protein degradation. And they also target genes responsive to environmental stresses, like superoxide dismutase, lactases, and ATP sulforylases. But the main, I would say, are transcription factors. A lot of them controls transcription factors. And we go further?
Yes, ma'am.
So now a little bit about development of and microRNA's target modules. So this is juvenile plant, Arabidopsis plant, going to adult plant. What is controlled by a module of microRNA 159 that targets SPL transcription factors, a group of SPL transcription factors, not all of them. And we are talking about Arabidopsis, but actually this module of 159 and SPL is quite conserved among plants. So and during juvenile phase, microRNA 159 is quite upregulated, leading to the downregulation of SPLs. And microRNA 172 is actually downregulated, leading to expression of apetalin-2. This is also a kind of transcription factor, but this is inhibitor of flowering. Yeah? And then when we have adult transition from vegetative to generative phase, microRNA 159 goes down, and SPL1 transcription factors go up, and they actually activate microRNA 172 gene expression, and microRNA 72 downregulates apetalin-2, and you get flowering phenotype. Of course, it is not so simple, but the transition is, for this transition, this is a critical role of this module of microRNA 159 and 72. And we have also other modules that are important for leaf morphogenesis, for shoot apical meristem maintenance, for root development, and so on. But this is, I think, the most studied and well-known example how transition from vegetative to generative phase is obtained. So microRNAs, as I said, also are responsive to different abiotic stresses. This is low temperature. So low temperature, we found that some of microRNAs may be downregulated. And for instance, in Arabidopsis, this is microRNA 169. Some members of this family are downregulated. Then you get upregulation of a given transcription factor, and you give upregulation of auxin biosynthetic genes. So auxins are increased. For instance, in potato, you have two different microRNAs that are downregulated, leading to the production of abscisic acid, so stress-responsive phytohormone, or leading to the production of gibberellins. And finally, in what we know in rice, you can get downregulation of 319, and you get the inhibition of production of reactive oxygen species. And these all responses in different plants may co-regulate enhanced cold tolerance. So knowing how microRNAs are expressed, we may also regulate expression of microRNAs in different varieties, obtaining plants better responding to a given environmental cues. So you can act at the level of microRNA, you can act at the level of target gene, but acting at target gene when they are transcription factors could be a little bit risky, because you may get a severe phenotype. It's much better to fine-tune microRNA expression to get better results. And microRNAs target modules in response to salt stresses. In some plants, several microRNAs are again upregulated, then they downregulate different targets, what leads to antioxidant production, ABA, abscisic acid response, and this is auxin signaling is improved, what also leads to molecular adaptation to salt tolerance. So you see that you may have different responses by microRNA downregulation, by microRNA upregulation, shaping the response at the level of a module or of a transcription factor or and further executor protein. So if microRNAs so strongly regulate different processes in development and responses to stresses, they themselves must be also very carefully regulated. What means that both deficiency and excess of microRNA may be deleterious in plant development. So this is a nice example about TCP transcription factors class 2, that negatively regulate cell divisions. So here you have a wild type Arabidopsis rosette leaves at the rosette leaf stage plant. Here you see a small seedling, natural wild type seedling. What you see here at the forefront should be like seedling, but it is arrested after a couple of days of growth. And why? Because TCP mRNA cannot bind microRNA 319. So what means that you mutagenize TCP mRNA in the region where it binds to microRNA. It cannot bind anymore 319, so it is upregulated. And it negatively regulates cell divisions, and plant stops at this stage of development. And in the background, what you see, there is too much of microRNA 319, so you have decrease in TCP mRNA level, causing excessive cell division in leaves, which gives them a cranky shape, like cabbage-like phenotype in comparison to wild-type plants. So this is a very nice example showing how microRNA level can affect development of plant, just single microRNA. 319 is actually really important microRNA. Okay. So now we are studying a lot about microRNA biogenesis and function. Today I will show you some results of our work on microRNA biogenesis regulation, and in the next lectures sometimes we will talk about our results where we will show you the role of some microRNAs, mainly in response to heat and stress and in response to drought. So as I mentioned, do I have it? Oh, it's off. I have no more. I think I will need it.
Which side would better?
Sorry? I can show here, of course. So we have a gene that is transcribed by RNase polymerase II. You know it already, RNA polymerase II. So you get prime microRNA, then DCL1 cleaves stem and loop structure and producing so-called precursor mRNA, and then DCL1 is producing microRNA star or passenger microRNA doublets. But the stem and loop structures may vary in their shape and structure between different species of microRNAs. So here are just examples of Brachypodium dictyosticium. This is a model plant for monocots. And you can see how different the stem and loop structures may look like. They may have microRNA just at the bottom of stem and loop structure. They may have it in the upper part of stem and loop structure. They may have it just in the middle of stem and loop structure. They may have it in opposite, in 5' arm, 3' arm. They may have it in a complete complementary way, while they may have partially be in a kind of open big loop of of course, these open loop, these are only bioinformatically designed secondary structures, what may mean that these big loop structures are not like fixed structures, because this is dynamic. Sometimes they are closed, sometimes they are opened. And you have also pay attention when you use programs for secondary structure design, that many programs by default have 36 degrees, what is not normal for plants, because for plants, at least in our country, is like 20 degrees. Here would be probably a different temperature, maybe 30 is more appropriate, but I think you have always remembered that to adapt the default temperature of a program to your conditions of plant you work with. So you may have really various structures of microRNA precursors. So, and now a little bit about DCL1 protein, because DCL1 protein, dicer-like 1 protein, belongs to RNase-3 type ribonucleases. What does it mean, RNase-3 type ribonucleases? So this is a single RNase-3, this is not a DCL1, this is RNase-3, that recognizes double-stranded RNA regions and keeps them leading to some sticky 2 nucleotides long 3' overhangs. So you have 2 single nucleotides at 3' and here, and 2 unpaired single nucleotides at 3' end. So a little bit like restriction enzymes in DNA, when they cleave, you get these cohesive ends. And here also in double-stranded RNA, it cleaves like leading to these 3' single overhangs, but only by 2 nucleotides. So this is typical for enzymes of the class RNase-3, and dicer-1-like, and also drosha and dicer in humans, all of them belong to RNase-3 family. So this is the dicer-1 protein. This is a quite big protein. It has almost 2,000 amino acids and has different domains. This is helicase domain. This is a domain of unknown function, so-called 283. Now it is known that this is a novel RNA binding domain. Then you have PAS domain, responsible for maintenance of these single-stranded 3' ends of stem and loop structure. And then you have RNase domain 1, RNase domain 2, and 2 domains for RNA binding. And when you look at the tertiary structure of protein, it has this kind of shape. You have a head, but you have this PAS domain for carefully maintaining in correct orientation of 3' end of single-stranded 3' end of precursor of microRNA. Then you have...
You have to use your finger because you cannot see.
Is better here?
Yeah.
Okay. Okay. So here is PAS domain, which binds to 3' single-stranded 3' end of stem and loop structure. Then you have 2 RNase 3 domains. So, 2 domains. This is why you can excise from stem and loop structure and to duplets of microRNA stem. One cleaves from one side and the second from the other side of stem and loop structure. And thank you very much. Of stem and loop structure. And then you have here helicases that allow to adopt proper structure of stem and loop structure embedded within DCL1 protein, and you have also this DAF domain that is novel RNA binding domain. We call it head, core, and base. And also recently, quite recently, a cryo-EM crystal structure of Arabidopsis thaliana DCL1 protein has been obtained, bound to a precursor of microRNA, which means we have no long primary transcript, but already cleaved stem and loop structure. So you can see here microRNA duplets embedded within the dicer-1 protein. Here you have RNase 3B, RNase 3A, these two RNase 3 domains, cleaving here and cleaving here, releasing duplets of microRNA and passenger microRNA. And then when you turn it by 180 degrees, you can see it even better, I think, here, that Pa's domain is maintaining this 3' 2 nucleotides overhang, and then you have these RNase 3A, RNase 3B, cleaving out the doublets of microRNA, microRNA star, and here you have these helicases and RNA binding domains that allow to adopt proper structure for cleavage for RNase domains. Okay, so as I have shown you, we have long precursors, we have short precursors, and it led to the discovery of two modes of microRNA biogenesis, so-called base to loop and loop to base. And BTL, I will call it LTB. So base to loop means that normally in plant precursors, you have a small loop, internal loop, and then it's like 15 nucleotides that are measured by DCL1 protein, and then you get first cleavage and second cleavage. So first cleavage is nearer base, and this is the mechanism of base to loop. This I will tell in a moment. And now the other one is loop to base. So it means that dicer-like 1 protein binds from the other side microRNA precursors. The first cleavage is near the loop, and the second cleavage is near the base. Yeah? So these are two mechanisms found, BTL and LTB. And since sometimes these stem loop structures are really long, they also there were discovered so-called mechanisms of sequential base to loop and sequential loop to base processing. So sequential base to loop, where you have here microRNA and microRNA star, means that you have this internal loop, 15 nucleotides, then first cleavages, and then second and third cleavage give rise to microRNA star doublets. So this is sequential base to loop. First you have cleave this part, and then the main part here. And in sequential loop to base is practically the same. You get first cleavage here, second cleavage here, but the third and fourth cleavage will give rise to proper doublets of microRNA star. So we have two modes approaching microRNA precursors, BTL and LTB. And this is not very clear from the beginning which one is for which precursors the right one. Some of the precursors may execute both modes of approach. Some are typical BTL, some are typical LTB. So this must be somehow checked experimentally. So now there was and there is still a discussion how the processing of microRNAs takes place. Whether it is during transcription already that microRNA precursor is cleaved and duplex is generated, and there are other researchers that tend to think that the transcript is then directed to so-called dicing bodies that are present in nucleus. So we asked the question, because in our former studies we found that splicing, because some precursors of microRNA primary transcripts contain introns. And we discovered that these introns are important for proper microRNA biogenesis. So if splicing is co-transcriptional, and it is no doubt co-transcriptional, then we thought that the processing should be also co-transcriptional of microRNA precursors. And we wanted to prove it, and in cooperation with Artur and with Pablo Manabella from Argentina, I think I will be able to provide you some proofs that it is at least partially co-transcriptional. So what we did, this is the gene for microRNA 163, and this is the precursor also, like it looks like before splicing. And this is the gene for microRNA 156A, and also the precursor, it looks like before splicing. And we designed probes for fish, fluorescence in situ hybridization. So probe is labeled fluorescently, and then you can, via microscopic studies, fluorescent microscopy, look where the probe can be found. And we did probes for exon, we did probe for exon-exon junction after splicing, and we did probe for intron. So by that way we can show precursors. Yeah? And the same for this gene, for exon, for intron, and for exon-exon junctions. And what we found during in situ hybridization, fluorescent in situ hybridization, that in the nucleus, prime microRNAs localized in one or two spots. So, for instance, with intron probe, we find it here, one spot. For prime microRNA 156A, one spot, and in control, of course, nothing. And in the case of prime microRNA 163, with intron we see here one spot, here we see two spots, here we see again one spot, and in control nothing. So 1, 2 spots means that when plant is diploid, sometimes both homologous loci from diploid plants can be active, sometimes only one. So this is why we see sometimes 1 or 2 spots. Clear? Is it clear? Yes. So we did the same experiment using RNAs. So we treated our specimen with RNAs to get rid of RNA, and of course we have no signal anywhere. What means that we really detected not DNA via hybridization, but RNA. Yeah? And then we wanted to know whether these spots of the precursors colocalize with RNA polymerase II, that is responsible for transcription of microRNA genes. So this is a probe which allows us to see prime microRNA 156A. Some experiments we did only for 156A because this is the longer one. It is easier to see if it is longer, if it's a longer precursor. And this is where you see in the nucleus dispersed dispersed presence of RNA polymerase II, labeled with not labeled, but we use fluorescently labeled antibodies against serine 5. Serine 5 is phosphorylated in the CTD domain of RNA polymerase II, and it is a signal that this is the polymerase is in the initiation state. Yes? Yeah? So using antibodies against serine 5 of CTD domain, we see only polymerase that initiates. And then if you merge this spot with this, you can easily see that it overlaps, and you can also measure it, and it overlaps, the spot and polymerase II. Then we wanted to know whether RNA polymerase II detected using antibodies against serine 2 of CTD domain, this is elongation, is also colocalizing with this spot. And this is at the edge, but actually you can see that it is an overlap. When you measure it, you see that the spot overlaps with RNA polymerase II. And then we wanted to know whether we see de novo transcripts. And we used bromouridine, and which is incorporated, of course, 3 nucleotides, which is incorporated when we add bromouridine 3 phosphate nucleosides, 3 phosphonucleosides to the probe. Only de novo transcripts will be visible. And you can use then antibodies against 5-bromo-U to detect transcripts, de novo transcripts. So what you see here is prime microRNA 156A, done with this probe I explained earlier. And then we looked for colocalization of these newly formed transcripts with our probe. And indeed, we found colocalization here. These are all three of them, colocalization. And when we measure in the straight line the colocalization, we see the clear colocalization of polymerase, RNA polymerase 2 with the spot for prime microRNA. So these are not spots of precursors that were once done, some were still colocalizing with RNA polymerase 2, but these are de novo transcripts where they are generated. So we wanted also to know whether the core elements of the microprocessor, like dicer-like 1 protein, like L01 protein, also colocalize with these spots we just proved are newly formed transcripts by RNA polymerase 2. So what we did, this is the spot for 156A, and this is the localization of L01. And L01 has localization which is dispersed within the nucleus, but it also can be formed in a specific foci, and L01 and DCL1 are markers for these so-called dicing bodies I mentioned earlier. Yeah? So when you look first at this dispersed form of L01 and our spot, you can colocalize it here. So L01 with prime microRNA 156 colocalizes when it is in its dispersed form. But now this is the same photo, but we paid more attention to this foci of where L01 is making this foci called dicing bodies. So here is the focus of dicing body, and here is our probe. So they do not colocalize. And this is prime microRNA, again 156A, and DCL1 that may also be dispersed. So these are nucleoli that DCL1, L1 polymerase 2 are excluded. Yeah? And this is in this dispersed form. Let us first check. This is our spot of microRNA precursor 156A, and we see that it colocalizes. While when we focus now in the same photo for DCL1 in these spots in dicing bodies, we see that they do not colocalize. So it was the first hint that newly transcribed microRNA genes colocalize with RNA pol2, colocalize with DCL1 and L1 that are not present in dicing bodies. So this is just dicing bodies. DCL1 and L1 are enriched in round nuclear bodies measuring 0.2 to 0.8 millimeters in diameter, as well as being diffusely distributed throughout the nucleoplasm. And the idea presented earlier by some researchers was that the transcripts are going to these dicing bodies where they are processed to mature microRNAs. So, arguing with this idea, we performed additional experiments, asking also a question: what is the function of these bodies? If we think it is not processing. So this is again that prime microRNAs are not detected in D bodies. So you can see here is microRNA precursor, primary transcript. Here is D body. So they are really apart. Here you see two spots of primary transcript of 156A, and these are D bodies. Here you see again 156A. This is precursor, this is D body. And this is for the other one, transcript of 163. D bodies are here. And here is the transcript. D bodies are here. Here are transcripts. D bodies are here. Here is transcript. So still we see that they do not overlap, transcripts and D bodies. So what we did, we inhibited transcription of RNA polymerase 2 with using alpha-amanitin, And we found that inhibition of transcription stimulates formation of antibodies. So you can see here prime microRNA 156 in PBS, this is a buffer, and here when we added alpha amanitin. So here you see the probe of prime microRNA 158A, which disappears after adding of alpha amanitin because transcription doesn't take place anymore. And what you can see here, you can still see when you merge the spot of when it's on the buffer, but there is nothing when you add alpha amanitin. But if you then look for the presence of I1 and DCL1, where they are now, whether more dispersed or more in the dicing bodies, here you see this dispersed formation, but here more and more dicing bodies appear when you add alpha amanitin. So it looks like D bodies probably may be storage places like Cajal bodies for splicing, for SNRPs. Like D bodies may be also a place where machinery for microRNA processing is stored when it is not needed. So we did another experiment that shows that inhibition of transcription by alpha amanitin stimulates formation of D bodies. So this is the presence of HI1 in root tip. And when you add alpha amanitin, it is already more condensed. But wait a moment, if you look at the in meristem in higher magnification, you can see HI1 in PBS that is rather dispersed, while here you can see the appearance of D bodies. And as we did the same as a control, we took M6A methyltransferase that is not involved in, let's say, in microprocessor, and we do not see any change of the localization of MTA between alpha amanitin and PBS. And this is just to show the whole cells, AGO1 that is cytoplasmic. You can see that there is no difference between AGO1 when you have PBS and alpha amanitin. Only for HI1 you can see the formation of these D bodies upon inhibition of transcription. So how prime microRNAs are processed then? Is prime microRNA processing coupled to transcription? So we have still two open possibilities. Although I just provided you data that strongly suggests that this is co-transcriptional, however, you must also prove it by another way, not only using one way. So this is microRNA gene with polymerase II and different transcription factors and mediator. Then you have elongation of the transcript. Then stem and loop structure is formed, and then probably it can be released from chromatin, and then you have post-transcriptional processing, or it is processed co-transcriptional attached to chromatin. So what we decided to do is the deep sequencing called PLANET. So this is plant native elongation transcript sequencing, PLANET. Yeah, so you isolate chromatin and you isolate newly formed transcripts, and then you sequence them to look for the 3' ends. And we were expecting to see different 3' ends in the case of precursors that will be a proof that dicer-like 1 protein cleaves already attached to chromatin. Is it clear? Or yes? Okay. So what we do, you isolate the nuclei, and then the chromatin isolation takes place from frozen tissue. So you isolate, after lysis of nuclei, the chromatin, the whole transcription machinery with attached RNA. Then you use, I forgot to say that you did everything in the background of a mutant where NRPB2-1 subunit of RNA polymerase II is deleted, but in that place this subunit is introduced, but it contains FLAG. So with FLAG you can immunoprecipitate, yeah, polymerase II complexes. So you isolate RNA polymerase II complexes, then using anti-FLAG antibodies attached to beads, you isolate the whole machinery, you immunoprecipitate the machinery, and then from the machinery, using proteases, you release RNA, and RNA is subjected to sequencing. And if there will be post-transcriptional processing, we expect to see only one cleavage, like that, the end of transcript. But if it should be co-transcriptional, we should see more ends of microRNA transcript after processing. So this should be like that you have stem and loop structure, that you may see here cleavage maybe here and here, if it is co-processing, co-transcription. So, yeah. So what we found is that planet data confirmed co-transcription and processing of plant prime microRNAs and revealed a difference in loop to base and base to loop processing modes. So this is, LTB prime microRNA processing from loop to base. Yeah. So what we see here, these are data from planet set. So we see 3' end here, 3' end enrichment here, and 3' end enrichment here. So this is this cleavage. This is this cleavage, and this is this cleavage. And if you look here, you see it? Do you see it here? Yes, so this is untouched transcript. Then what you can see here is that you see this violet one, this is this one. This loop, stem part of stem and loop is released, because this was the first cut in LTB. Do you remember?
Yes.
Yeah? So this was the first cut. And then what we see further, we see this part and this part. So this is the yellow one, so this cut. And we see also this one, what means this is already doublets of microRNA star, which is already, should be released, but probably is attached still to microprocessor that is at chromatin locus. That is why we see it still. So this is co-transcriptional. And this is LTBS, so sequential LTB. So what we see this cut, we see this cut, we see this cut, and this cut. Yeah, this one. So again, when you cleave it, so the first cleavage is here. So this is it green? I think it is.
Yes.
It is green. So this will be this one. So this is this cleavage, and the stem and loop is released. Then we see the violet one. So this is this, yeah, part. So this first sequential stem and loop structure is released. And then what we see this end. So this is here, the blue one. And we see again this duplex of microRNA star should be released, but is still somehow attached to chromatin. So this is in the case of LTB, what we found. And in the case of planet with base to loop processing modes, so what we saw, this is that we see BTL pre-miRNA processing, we see the cleavage here, and we see the cleavage here. So it means that it is cleaved, the stem and loop structure is cleaved, but then we lose it. We do not see it anymore. So what we see is here, we see this cleavage, yeah, here, and this cleavage. But we do not see these cleavages. So if we do not see it, we cannot say nothing. It can be co-transcriptional, but who knows, maybe this whole stem and loop structure containing microRNA star is processed in lysine bodies. That we still do not know. We cannot exclude this possibility. Although we were never able to detect microRNA precursors in the bodies. And the same is with sequential BTL, that we see just these primary cleavages releasing stem and loop structure, and we do not see it anymore because it is not attached to chromatin. So, in conclusions, we can say, are all prime microRNAs processed co-transcriptionally? I think I provided you data that LTB and sequential LTB are processed co-transcriptionally, we show you this microscopic data and planet data. So we have transcription, initiation, elongation, stem and loop structure, and then processing, co-transcriptional, at least in the case of LTBs, that approaches from up and then everything is touched to chromatin. And who knows what is about these BTLs that we cannot give still a proper answer. And this was done, the work with Arthur, with his PhD student, and in cooperation with Pablo Manavela from Argentina, recently moved to Malaga, I think, yeah? Malaga in Spain. And with this, I would like to thank you, and I will take questions. I think I spoke maybe even too long. Yeah? Too long?
No,
Too long, too long.
Too long?
I can say the truth.
So questions? Was it clear? Yes. But no question. Okay, so if you want me to ask, I am here around, and Arthur as well, and we can answer all your questions you want to, right? So thank you very much.
All this wonderful representation were done by PhD students.
Yes. So you will now have tutorials, yes? Sorry?
You said the work, this is
I think it's okay.
Or other?
So, good afternoon to all present here. Interaction of long non-coding RNA and microRNA in plants. So, microRNA can target the genes as well as recently it is discovered that it can also target other regulatory RNA molecules like long non-coding RNAs. So, I am working on that. Today we will basically cover some very basic points and basic databases through which we can download microRNA sequences, mature microRNA stem loop structures. So today is just an introductory part. So as covered by Zofia ma'am, very well covered. So microRNA are small non-coding RNA that regulate gene expression at post-transcriptional level. So it is first identified in 1993 in Caenorhabditis elegans, later found in plant, animal and viruses. So microRNA are essential for controlling various processes including growth, development, stress responses and gene resistance by regulating gene function. Basically these microRNAs cannot directly impart some resistance, some developmental function. They only impart through a gene because they target gene, that particular gene, like some hormonal genes like jasmonic acid, salicylic acid. These are the main hormones that provide biotic stress resistance. ABA that provide abiotic ethylene. Hai na? So the genes which are controlling these hormonal regulations, these genes are targeted by microRNAs. So basically microRNA function through gene, not directly. Okay? MicroRNA biogenesis overview. First is, this is a diagram made by me in my book chapter. So first of all, RNA polymerase which transcribe the microRNA through mid genes. Then the primary microRNA, as this is well explained by Zofia ma'am, as you can see on the slide. This primary microRNA is further processed by various proteins, and then precursor microRNA is formed. Then microRNA duplex is, so this whole process occur in nucleus. So by hasty protein, this microRNA duplex is exported to the cytoplasm. Then this microRNA duplex contains two strands, passenger strand and guide strand. Passenger strand which usually degrades, and guide strand do its further function. Then upon guide strand, various proteins like RNA induced silencing complex and argonaute proteins, they come and they help that guide strand to degrade the mRNA which is of gene. mRNA, and then after mRNA cleavage, now after mRNA cleavage, it can undergo 100% cleavage, or it can make transacting small RNAs. These transacting small RNAs further degrade other genes, matlab at secondary level. MicroRNA itself degrades some genes. After degradation of that gene, transacting small microRNAs, they further degrade other genes. Matlab it works at two levels. So these dicer-like proteins are key in processing primary microRNA to precursor and later into mature duplex. Argonaute protein is the mature microRNA guide strand is loaded onto argonaute protein forming RISC complex, and further is gene silencing. The RISC complex guides the microRNA to the target messenger RNA, leading to either degradation or translational repression. Mechanism of microRNA mediated gene silencing. Now how a microRNA basically recognize its target on a gene, on a mRNA. MicroRNA recognize their target mRNAs through sequence complementarity. and that sequence complementarity to a specific part of a gene, and that part is 3' UTR, 3' untranslated region, which is specifically recognized by microRNA to target the gene, to basically repress its expression. Is it okay or not? It is very basic. Then is modes of action. There are two modes of action: translational repression and mRNA degradation. Translational repression is the miRNA risk complex inhibit the translation, further protein formation of the target mRNA. And mRNA degradation is that previously I have shown in the diagram. In some cases, microRNA promote degradation of target mRNA, reducing its level in the cell. Now, these are the main databases of microRNA related to the plants. Okay? First is miRBase, which is widely used. Then is PMRD, Plant MicroRNA Database, PMIREN, Plant MicroRNA Encyclopedia, and miRNest. Today we will cover miRBase because it is widely used and more authenticated. miRBase. हाँ, miRBase के लिए लैपटॉप्स मतलब लेकर आए हो सब लोग? वाईफाई का पासवर्ड चाहिए किसी को? प्रोवाइडेड है या नहीं? वाईफाई का तो चाहिए होगा। आयना मैम को पूछना पड़ेगा। नहीं हम लोग दे देंगे, बट कितने लोगों को चाहिए? सब कुछ वाईफाई पासवर्ड मतलब अपना नेट से कनेक्ट कर दोगे? वैसे इट्स अ वेरी बेसिक डाटाबेस, फोन के उससे भी चल जाएगा, वो कनेक्ट करके देख लो। मैम हैंड्स ऑन के लिए जिन बच्चों के पास पीयू वाईफाई है वो तो शेयरिंग में कर लो और बाकी फोन से देख लो एक बार। हो गया कनेक्ट? हो गया सबका? जो भी पीयू वाले हैं वो हेल्प कर दो बाकियों की। हो गया सबका? नहीं मेरा नहीं है, मेरे को पासवर्ड नहीं याद। मेरा लैपटॉप दे दो किसी को। मेरा फोन आ रहा है मुझे लग रहा है। हो गया सबका कनेक्ट? ये जैसे पकड़ा, ये पकड़ ले। Now you all can search mir base. इस पे क्या पासवर्ड? इस पे तो खुद मांग रहा है।
तू अपनी मेल खोल के देख फोन में, उसमें पासवर्ड आईडी होगी।
नहीं मैं लैपटॉप ही अपना कनेक्ट कर लेती हूँ।
नहीं कहाँ करेंगी, मेल में चेक।
रुक जाओ, ये सर का लैपटॉप है,
पता नहीं।
अपना ही कर लेती हूँ।
लैपटॉप को कनेक्ट करना पड़ता कनेक्शन ही आ जाए।
अच्छा
पासवर्ड नहीं है।
एक मिनट ले। Have you all opened this page? Now you can go to search. As you can see, we can search microRNA by various means, like you can also use keyword, name of the species and specific microRNA accession. You can also do as you can मतलब साथ साथ में आप भी कर सकते हो, ठीक है? This is VV, like Vitis vinifera. This is the name of microRNA. First we can cover some basics, then you can all do it easily. So in miRBase, it is a searchable database of published microRNA sequences and annotation. Each entry in miRBase sequence database represents a predicted hairpin portion of the microRNA transcript. First we will just go through the basics of miRBase. Termed mir in the database with information on the location and sequence of mature microRNA sequence termed mir. Basically in miRBase, the mature microRNA sequence is termed as mir. Then after it, like 156, it's the number of the microRNA, number of its the exploration, whether the number of its research. Both hairpin and mature sequences are available for searching and browsing, and entries can also be retrieved by name, keyword, references and annotation. All sequence and annotation data are also available for download. You can also download all these. We will see later. How to use miRBase to search. Achha. How to use miRBase to search known microRNAs. You can search for on the search page, as we have gone on the search page. First we have gone on the search page. Now on search page you can search the microRNA by identifier, like I have also added like VV mir. Mir is basically the name of the microRNA and the number itself is the included in the name. Then keyword like elegans, the name of the species. By precursor sequences, you can also search the microRNA by adding its precursor sequences. By adding the precursor sequences, you can download the mature sequences of microRNA. Then by mature miRNA, choose BLAST-N to search for a microRNA in a longer sequence. Now, to search all microRNA of a particular organism, browse the database of microRNA. Then we will go on browse. In browse, you can just choose the species like Vitis vinifera, Cinorhabditis elegans. Just by clicking on the species, you can get all the mature microRNAs of that particular species. There you can access microRNA of various organisms. Now, what is the nomenclature conventions followed by miRBase? First is the numbering of microRNA genes is simply sequential. The latest number of microRNA shows the number of microRNAs identified in the organism, that is basis upon its identification. Like 156, it is identified at number 156 in that organism. And that similar sequences which are found in other organisms are also given the name 156. Like in grapes, it is 156, in Arabidopsis it is 156 because of the sequence similarity. Then the first three letters signify the organism, like OSA, mir 159. OSA denotes Oryza sativa. Like I have put in the search part VV. VV means Vitis vinifera. So here also OSA denotes Oryza sativa. So first three letters are of the organism, then MIR, that is commonly used for all microRNA, then is number. That number is based upon its identification. अगर sequence same है तो sequence that is upon sequence similarity.
How much percentage you have the sequence similarity?
Like I have personally searched various microRNA sequences. Like I have searched in grapes and in Arabidopsis. I have personally seen that there is 100% sequence similarity in grapes as well as in Arabidopsis.
तो तभी हम सेम नाम देंगे मैम, अगर 100% सिमिलैरिटी
In Arabidopsis, like Arabidopsis thaliana, ATH MIR 156. In grapes, that is VV 156. VV MIR 156. So different on the basis of the species, but MIR 156 is same in both. Mature microRNA sequence is same.
अगर 100% सिमिलैरिटी नहीं होगी, सिमिलर होगी, थोड़े बहुत मिसमैच भी हो जाते हैं, तो मैम अगर 90% हो और 10% मिसमैच हो तो भी सेम नंबर जाएगा कि तब अलग जाएगा मतलब जो miRNA उसका नंबर?
जो मैच्योर माइक्रोआरएनए है, अगर वो डिफरेंट है, अगर उसमें कुछ भी डिफरेंट है, अगर इवन उसमें आर्म्स भी डिफरेंट हो जाते हैं, वो आगे आएगा तो उसका डिफरेंट नेम चला जाता है. लेकिन अगर मैच्योर, आई एम टॉकिंग अबाउट मैच्योर माइक्रोआरएनए, मैच्योर माइक्रोआरएनए का अगर 100% सीक्वेंस सिमिलर है तभी वो उसका नाम जाता है. वो आप आगे देखोगे कि एक माइक्रोआरएनए में भी दो आगे नाम चले जाते हैं, दैट इज 3P और 5P. We will also study that what is the basis of 3P and 5P in similar microRNA. Next is lettered suffixes denote closely related mature sequences. For example, HSA MIR 121A and HSA MIR 121B would be expressed from precursors. Now this A and B is based upon the sequence similarity of the precursors. Precursors HSA MIR 121A and HSA MIR 121B respectively, because the precursors of these microRNA, these mature microRNA are similar to each other, and they are further processed from these precursors to mature microRNA. That's why they have given the name A and B. This is the basis of nomenclature A and B. Further, to denote the source arm of stem loop of. Now the precursor contains stem loop structure. That stem loop contains double-stranded loop, double-stranded, and that double-stranded contains two strands, 3' and 5'. If the guide strand is originated from 3' arm, then it is given the name 3P. If the guide strand is formed from 5' arm, then it is given the name 5P. Have you understood this? For example, MIR 142 5P from the 5' arm. That 5' arm gives the guide strand there.